Alright, this is going to be the section that talks about genetics. We're going to look at the differences between DNA and RNA. We're going to look at how DNA is used to make proteins. We're going to look at how cells reproduce. We're going to look at Mendel's law and the genetics that come from Mendel's law and like inheritance patterns and things like that, along with some applications of DNA technology and such. So this covers a quite broad topic that we covered in significant depth. Okay, so first of all, we're going to talk about the difference between DNA and RNA. And there are really three big differences. You have deoxyribose in DNA, so that is the main sugar there, whereas in RNA you have ribose. So the sugar difference is the first one and the main difference. DNA is a double helix or is double stranded, whereas RNA is single stranded. And also in their jobs. So DNA holds the instructions for the cell for what proteins need to be made, and then RNA helps in making those proteins and carrying out the instructions. So there's four bases in DNA. There's C, G, A, and T, and then in RNA there are basically the same four bases, except for instead of T you have U or uracil. So there's a big difference there when it comes to pairing, and we'll get into that in a little bit. Alright, so this is just showing structurally in a picture the differences. So like we mentioned, that DNA is double-stranded, where RNA is single-stranded. You've got the bases on each side of the image. So on the left there you've got the bases for DNA, on the right you have the bases for RNA, and all of them are the same except for uracil, which is right here. That takes the place of thymine or T in DNA. Alright, so another important part that DNA plays is holding the instructions for protein synthesis, or making proteins. So the general idea about proteins, or about information in the cell, is that we go from the DNA in the nucleus to RNA, which kind of acts as an in-between to protein. Alright, and there's two big processes we're going to talk about. You have transcription, which is where you go from DNA to RNA, and then you have translation, which is where you go from RNA to protein. And so this is what we call the dogma, central dogma, and this is how the genetic information is transferred from the DNA into proteins. Alright, so like I mentioned, transcription, we're going from DNA to RNA, and you're making mRNA here. So mRNA is messenger RNA. It carries the information from the DNA to the ribosome, which we'll talk about in a second. And a key important thing about this is this takes place in the nucleus, because that's where the DNA is found. Translation, on the other hand, is where we take that mRNA and make it into a protein. Okay, this takes place in the cytoplasm, in the ribosome. Okay, so this takes place in the ribosome, and the ribosome may be free floating, like just floating around inside cytosol or cytoplasm, or it can be attached to the ER, particularly the rough ER. And so the mRNA helps make the protein by combining with the ribosome, and the tRNA, which is transfer RNA, brings over the amino acids, and they come together to make a protein. And the third type of RNA that we've talked about, in class, is rRNA, and that makes up the ribosome itself. So mRNA is your messenger RNA, tRNA is your transfer RNA, it's responsible for bringing the amino acids to the ribosome, and then rRNA is what makes up the ribosome. Alright, so this is just an illustration, a little bit more detail of how translation happens, and I basically just kind of described it but this is a good illustration if you wanted to look at a picture of it. Alright, so how do we actually know how we go from DNA to RNA to protein? Alright, so let's kind of talk about a few things first of all. So first of all, we got to talk about the base pairing. Okay, so base pairing basically means what bases pair with what other bases. So if we're going from DNA to RNA, there are 4 combinations we can possibly have. So if you have a T in DNA, you would have an A in the mRNA. If you have a G in the DNA, you would have a C in the RNA. Vice versa, if you have a C in the DNA, you would have a G in the RNA. And the one that's unique is if you have an A in the DNA, you put a U for the RNA, because there is no thymine, there's no T in RNA. So we replace that with U. And so the first part of looking at how DNA goes to protein is translating the, or is transcribing, excuse me, the DNA into RNA by using these base pairings. And you have to know these in order to do this process. Then what we use once we get the RNA is what we call the genetic code, okay? And that's what this chart is here. The genetic code relates the RNA sequence to proteins. Alright, so if we look at here, we've gone from DNA to RNA using that base pairing, and then we can take the RNA sequence and go to amino acids. And the way you do this is the first amino acid, or the first base is on the left here. The second base is going to be the columns. So the first base is the rows. I should say that a little bit clearer. The second base is the columns, and then the third base is going to be the individual rows within each box here. Okay, so you go basically, if we're going to look at this first triplet here, you're going to go U, which is going to be this row, C, which is this column, so we're looking in this box here, and then you're going to look at the second row within the box, which is going to be SER, or serine. Okay, so it basically is kind of a coordination of coming together. You find the row, you find the column, and then you find the row within the box that you've isolated to get that third letter. Alright, so there's an example here that's done, but the one that's in your study guide we're going to do together real quick. Okay, so you start out with DNA, and the DNA they give you is TTA, CGA, TCA, ATG. Okay, so we can use those base pairing to get the RNA. Okay, so you are going to do AAU, because that's what pairs up with those, GCU, AGU, and then UAC. Alright, so AAU, GCU, AGU, UAC. Alright, so what we're going to do from here is we're going to need to go back and look at that amino acid chart that we were looking at a minute ago to figure out what these actual amino acids are. Alright, so let's actually go back and look at that real quick. We're going to go back. Alright, so we're going to take that sequence we had. Let's clear everything off the screen here. We're going to take that sequence. Now this is RNA. Okay, so this is the RNA sequence that we had, which was AAU, GCU, AGU, UAC. Okay, I want to make sure that I copied that correctly. Yes, I did. Alright, so now to get the amino acid sequence. Okay, so we're going to do each of one of these in turn. So we're going to do A, so it's going to put us in this box here, and then U, which is the first column, that's going to be ASN. Alright, so we can get rid of that little thing that we drew there. Then we're going to do G, okay, so that's GCU, C, we're going to go here, so this box here, so all of them, and then we're going to do that first column. It doesn't really matter because they're all basically the same, ALA. Okay. And then we're going to do AG, okay, so that's going to be these guys here. And then we're going to look at U, which is the top one, so that's SER. Okay, did not mean to do that. Okay, so we had our RNA. Again, let's go redo this real quick. AAU, GCU, AGU, UAC, and our amino acid sequence so far was ASN, ALA, SER. Our last one, okay, was UAC, so we're going to do U and A, and then the second one here, the second row there is TYR. Okay, so that's the amino acid sequence given the DNA sequence that's on your study guide. Okay, so you don't have to memorize this codon chart. You do have to be able to use it on a test. You will be given it to it. Okay, and the reason why it's called a codon, I need to mention that, is that if you notice, I put them in groups of 3. But basically, every 3 bases, so every 3 bases equal 1 amino acid. Okay, and that's what the codon chart shows you, is that for the 3 bases, what 1 amino acid does that equal? Alright, so that is protein synthesis, and that's very important, but it's also very important that DNA be able to copy itself, particularly when the cell gets ready to copy. And the whole process of DNA replication is when DNA copies itself. And there's one key thing to know from this process, okay? The big thing that you need to know is what actually does it. It's called DNA polymerase, alright? And this is the enzyme that actually creates the new DNA, okay? And if you think about DNA as a polymer, polymerase is the enzyme that makes that polymer. And the thing about this that's really important is we call this semi-conservative. What that means is that you actually will use the 2 DNA strands that already exist to create the new copies. Okay, so the original strands are going to be used as templates, and so when you create 2 new copies, or when you create your new copies of DNA, you're going to have 1 original strand and 1 old strand. So you're semi-conserving the original DNA. Okay, so we call that semi-conservative. Now, why is DNA replication important? Because that leads us into cell division. Okay, so the first type of cell division we're going to talk about is mitosis. And mitosis' job or function is to create new cells when cells get worn out. Now, the thing about mitosis is a couple of things. This is asexual reproduction. Okay, you're basically making a clone of your original cell. Alright, this is going to happen in what we call somatic cells. These are basically your body cells, so anything that's not an egg or sperm. And they are what we call diploid. That means you have a full set of chromosomes or 2 copies of the chromosome. So think about diploid, di means 2. So for humans, you're going to have 46 chromosomes or 23 pairs. So you'll start in the original cell with 46 and you end with 46. And the process of this, the acronym I used a lot in class was what we call PMAT. Okay, so let me make sure I can actually write this correctly. So PMAT will help you remember what the different stages are. So PMAT, so prophase, metaphase, anaphase, and telophase. So prophase, basically you are breaking up the nucleus, and you're condensing the DNA. Okay, but the big thing is the nucleus breaks up. Metaphase, they line up in the middle, they being the chromosomes. So the chromosomes are going to line up in the middle. Anaphase, they're pulled apart. The chromosomes are pulled apart. And then finally in telophase, the nucleus starts to reform. And Cytokinesis, this is something that's not necessarily part of the whole PMAT thing. It happens basically at the same time as telophase. Cytokinesis is when the cell actually splits. Okay? So that's mitosis. The other process we talked about is meiosis. This is very different than mitosis in the fact that you're actually going to do 2 replications here. Okay, you still go through the same phases, but there are a variety of differences. First of all, the purpose of meiosis is to make the gametes, the sex cells, the egg and sperm. And they are going to be what we call haploid. They only have 1 copy of every chromosome, which means that they have half the number of chromosomes. And another thing here is we have something known as crossing over. This is where you have parts of the chromosomes swapping sections, and the reason we do this is to create genetic variation. You create different parts. And so what happens here is you start with 1 cell up at the top, and you end up with 4 at the bottom, because you go through PMAT, the same PMAT as you did before, with twice. Okay? And so the reason why we need these is because we need to create those eggs and the sperm. And so if you think about this, if the sperm has 23 chromosomes and an egg has 23 chromosomes, when you put them together, you get your 46. Okay? So if they only had 46 and you combine them together, you would end up with 92. Okay? So we have to create half the number of chromosomes in order for when the egg and sperm come together to create a fertilized egg to have the right number of chromosomes. Alright, so mitosis is just for repair and create new cells. It's a clone. And meiosis is to create the gametes. So meiosis is involved in sexual reproduction. Alright, so now I've talked about asexual and sexual reproduction. Let's talk about why they're important, okay? Or what the advantages of each are. Okay, so for asexual reproduction, you don't need an egg or sperm, whereas with sexual reproduction, you do. Okay, so that can be a good thing, that can be a bad thing. You create clones with asexual reproduction, but you get a lot of diversity, which is a really good thing for like evolution and stuff like that. So sexual reproduction, a good thing about that is that it creates diversity or variation. Asexual reproduction happens very quickly, whereas with sexual reproduction, you have to have both a male and female, or have the egg and sperm, which can take longer. So there is good parts to both. There are bad parts to both also, but generally, you know, there is a reason for each one of them. Alright, now one thing, there's a lot of information on this slide, but the big thing I want to point out is we have a couple of special terms, okay? So the special term for sexual reproduction, or excuse me, asexual reproduction in things like bacteria is binary fission. Okay, this is going to happen in bacteria and protists. It's very similar to mitosis. It's a little bit simplified. And then conjugation is the sexual reproduction for bacteria and protists also, but they don't have egg and sperm. They just exchange DNA pieces. And so it helps them get that genetic diversity that otherwise you would get from sexual reproduction. So there are special terms for the sexual and asexual reproduction in protists and bacteria. And so it's just important to know what those terms are. Alright, so now we're going to get into some of the inheritance stuff. And so, like a lot of things in biology, it's good to understand what exactly some of these terms mean. So to start with, let's talk about genes and alleles, okay? So your gene, let's say we're looking at the gene for flower color, like on a plant or something like that. The allele is going to be the different versions of it, okay? So if it's blue or green or yellow or pink, the allele is the different versions. The gene is the characteristic you're looking at, whereas the allele is the different versions. And then there's 2 ways you can talk about it. You have the phenotype, which is the physical description, like what does it look like? And then you have the genotype, which is what are the genes, okay? So what genes are there to represent them? A lot of times we represent these with different letters, okay? And so in the genotype, we'll talk about the different types of allele. And we can break them into 2 categories. And it's very, very easy. We have dominant alleles Okay, these are the ones that show up the most often. They are represented by a capital letter, and they will mask the recessive trait. Thus, they show up more often. And then you have the recessive allele. Okay, these are going to be less common, and they are shown with a lowercase letter. Okay, and so they get masked by the dominant allele. Okay, and we have a couple of different ways we can represent the genotypes based on what type of alleles they have. A heterozygous genotype means that you have one of both. Okay, so that's one big letter and one little letter, like shown here. And this is what I mean. Because you have one of both, it shows the dominant phenotype. So it shows the dominant trait because the dominant allele will overpower it. Then you have homozygous, which means the same allele. You have homozygous dominant, which is two big letters, and then you have homozygous recessive, which is two little letters. Okay, so you have the same allele, and it just depends on whether it's recessive or dominant, what trait they show. All right, now we're going to start getting into Mendel, and he had three big ideas. The first is what we just talked about, the rule of dominance. That there's always going to be a dominant allele, and there's always going to be a recessive allele. Then he talked about the law of segregation, and this is going into meiosis. He's talking about how when you have pairs of alleles on these chromosomes, that they are going to be separated. That each gamete, or each egg or sperm, will end up with one copy of, or one allele, and that they're separated to, create diversity. And then finally you have the law of independent assortment, and this talks about the genes itself, that the genes are not going to be related, that they are sorted independently. So if you look at this image over here on the right, you'll see that we have big G and little g, and big Y and little y, and the big letters can all go together, the little letters can all go together, which is the top situation that they show you there, or they can be mixed up. So like in the bottom situation, you get the big G's and little Y's, and vice versa on the bottom. So they're randomly mixed up. They're not linked together, is what that's saying. All right, so how did this lead us to looking at inheritance? The biggest tool when we're looking at inheritance is a Punnett square. And the purpose of the Punnett square is to show us what offspring could we possibly get from what we call a genetic cross. When we mate two things, we look at their offspring, we can talk about percentages, we can talk about ratios, and we can talk about it as either the phenotype or the genotype. Remember, the phenotype is what they physically see, whereas the genotype are the genes. Alright, so now we're going to move into things that don't fit into Mendel's ideas. Okay, so Mendel was correct in a lot of things in what he said, but since then we've seen that there are other types of inheritance patterns. Two of the biggest ones are going to be codominance and incomplete dominance. And there's a quick way to kind of show you how these work. Codominance is when both are dominant. Okay, so if you see here in this picture on the upper right, you have a black chicken and a white chicken. It gives you what we call a checker chicken. Okay, and so with codominance, the heterozygous one, the one that has both a big and a little one, is going to be checkered pattern or spotted. So both colors show up. So that's kind of how you know, if they describe something along those lines, that's how you know it's codominance. Whereas with incomplete dominance is where you don't have either one be completely dominant. So in the lower right there, you have a red and white, and you get pink. So with this one, the heterozygous, okay, so for the one down here, the heterozygous is going to be a blend or a mix of the two parents. Okay, so if you had a tall and short, then the offspring that had both letters, the heterozygous one, would be medium. So it's going to be somewhere in the middle. Alright, we also have what we call multiple alleles. This is actually technically a type of codominance. This is blood typing. So A, B, and O. In this case, you have three, or you have two alleles, or excuse me, more than two alleles. You have A, B, and O that you can get, and the combination will give you the blood types. With polygenic traits, this is something that doesn't really look at alleles or whatever, this is where you have things that will show up on a spectrum. Eye color, hair color, skin color, things of those nature. They're controlled by more than one gene. So your skin color is controlled by multiple genes. That's why you get such a range in color, from very, very pale white to very, very dark skin. We call that polygenic. And the way you can remember it is poly means many, and then genic means gene, or sounds like gene. So many genes. That's a really quick way to kind of remember that. All right, now getting into some of the last bits that we talked about with genetics, which was sex-linked traits. But first of all, we need to talk about the chromosomes. We haven't touched this yet, but you have 23 pairs of chromosomes in humans. The first 22 pairs are what we call autosomes. They have most all the traits except for the gender. Okay, the sex chromosome, which is the 23rd pair, deals with the gender. Okay, so that's important to remember that it's the 23rd or the very last pair. Okay, so you have two genders. You have XX, which is a girl, XY, which is a boy. All right, and the way this works is that the females will donate the X's, and then the males will either have an X or a Y in their sperm. So it really is the males who determine the sex of the baby based on what is added to the egg. Alright, and so sex-linked traits are going to be those traits that are carried on the sex chromosome, specifically the X. There are traits on the Y, there's just not enough about them that we're going to focus on them. So we're going to focus on the X chromosomes. Okay, so sometimes they're called X-linked, and with these, it's very important to realize that boys are going to get them more often. Okay, so boys are XY. They're either going to be dominant or recessive. They only have one X, so they can't be heterozygous. A lot of these traits are recessive, so for females, because girls are XX, they have a second X that can help balance out the recessive trait. And the only way they can get the recessive trait is if they get both recessive alleles. Where with boys, they only have one X chromosome, so they either have it or they don't. Also, girls can be what we call carriers. And carriers basically mean they are heterozygous. They don't have the trait. Like I said, most of these are recessive traits, but they can pass it on to their offspring. So some examples I show here are color blindness and hemophilia. Hemophilia is when you don't have the ability to clot blood. Alright, the other thing we talked about was a pedigree. Okay, a pedigree is used to show like inheritance patterns. Okay, so you can typically the circles are females, or not typically, circles are females, squares are males. If they're colored in, that means they have whatever you're dealing with. And so it looks like a family tree, but it typically will have colored in portions to show the inheritance. Alright, and then finally we're going to wrap up with some disorders that can come from either mutations or from improper meiosis. Okay, so the first one is non-disjunction. Okay, non-disjunction is the general term for when something doesn't have the correct number of chromosomes. Okay, so it can be too many or too few. An example is Down syndrome, where you have too many at the 21st chromosome. So if you look in the upper right corner here, this image shows that the first two cells would end up with one extra one, and the bottom two cells end up with one less. So like I said, it can be too many or too few. And then you have two general types of DNA mutations. You have frameshift mutations. So if you're looking at a triplet, okay, we call this a codon, and we call it a reading frame. Basically the fact that you read every three codons, kind of shown here in the lower corner. If we have a frameshift mutation, that means that you have removed something or deleted or inserted something where you disrupt the reading frame. So now it's changed how we read those three base pairs, and that can change what amino acids there are. On the other hand, you have point mutations, which are where you simply just switch out a base. You change an A for a C, or a G to a T, whatever. But it's a single point. It can still affect if the amino acid is changed, but it's not as disastrous sometimes. Alright, this is just some genetic disorders that we talked about. It's really not important to necessarily know what these disorders are. Just know that a lot of times we can find them out by what we call a karyotype. And a karyotype means that you take all of the chromosomes of an organism, or like a baby or something, and we put them out and we look at them and make sure that they look right, that you have the same number of them. It's a visual way to inspect the chromosomes. Alright, and then finally we're going to finish with some of the technology that has come out of DNA and using DNA. The first is what we call DNA fingerprinting. This is used to basically compare DNA. Okay, so you can look at a suspect, you can look at paternity, you can try to find missing people, and it's always a comparison, whether they match or not. And then genetic engineering is a huge part of almost our daily lives in a way, and this is where we're modifying DNA in a organism to do something we want. A lot of times we're putting in a trait we want. It can help us make food, it can help make food better, and the DNA that we use with this is called recombinant DNA. It has something that's not natural, like that's not naturally in there. We've stuck something in there. Alright, so this will wrap up our section on genetics. It is a bit of a longer section just because it covers so much detail, but it hopefully will make a little bit more sense now.